Dear friends, in today's lecture, we will try to cover the sexual reproduction, structure of antheridium, structure of archegonium, fertilization, alternation and generation in the Anthoceros. In our first part of Anthoceros life cycle, we covered its classification, occurrence, number of species, then external characteristic of the plant body, then anatomy of the thallus, then vegetative reproduction, and after vegetative reproduction, we are starting here with the sexual reproduction. Okay, so sexual reproduction in Anthoceros. Point number 1. Species of Anthoceros are both monoecious and dioecious. Monoecious means what? Male plant body and female plant body different. Male plant body produce the antheridium, female plant body produce the archegonium. And in case of dioecious condition, the male reproductive organ and female reproductive organ both are present on the same plant body. So, in case of Anthoceros, sexual differentiation is the both types, monoecious as well as the dioecious. Point number 2. Both male and female sex organs are embedded म्हणजे असे रुतलेले. Both male and female sex organs are embedded in the dorsal surface of the side. Dorsal surface means upper surface of the thallus. Okay, the male antheridium and female archegonium both are embedded toward the upper side or dorsal side of the thallus. Number 3. They develop just behind the growing apical regions. That is the position where they are present. They develop just behind the growing apical regions. Okay. The monoecious species are protandrous, means their antheridia matures before archegonium. When the Anthoceros species are monoecious, such species are called as the protandrous because in which the antheridia mature first before the archegonium. Now, structure of the antheridium. Point number 1. The antheridia are present in the antheridial cavity. In case of Hepaticopsida and Anthoceratopsida. In case of Anthoceros, the antheridia are always present in the antheridial chamber or called as the antheridial cavities. Okay, the antheridia are present in the antheridial cavity or antheridial chamber with a sterile roof of 2 to 3 layers, and the wall of that antheridial chamber is 2 to 3 layered. Okay. Number 2. The mature antheridium is a stalked. Mature antheridium having the small multicellular stalk. Then it is club shaped structure. This antheridium is club shaped structure. Okay, structure of the antheridium is club shaped. Then with single layered jacket. That antheridium further covered by the single jacket layer, which is the sterile jacket layer, which is the protective covering of the antheridium. Each cell of the jacket possesses a prominent plastid, and each cell of the jacket having the prominent plastid material. Okay. Number 3. Inside the jacket there are large number of androcytes. Inside the jacket there are large number of androcytes. See here the structure of matured antheridium. Matured antheridium having the small stalk. Then body is there. Body cells are called as androcyte mother cell or androgonal mother cell, and each cell of the antheridium, each androcyte mother cell produce the single sperm. So what is the function of antheridium? Antheridium produces many sperms, and these sperms are nothing but the male gametes, and each sperm having the single male nucleus which is haploid in nature. And in case of Anthoceros or in case of bryophytes, the sperm is the biflagellated structure. So this is the biflagellated sperm. Okay. Now this sperm also called as spermatozoid or antherozoid or male gametes. So in this way many male gametes are produced by the antheridia. Okay. Now the structure of the archegonium. The archegonia are embedded in the thallus and only the cover cells project. Only cover cells project beyond the general surface of the thallus. Toward the dorsal side of the thallus, the archegonia are embedded and only cover cells are projected beyond the upper epidermis. Okay, cover cell means that is the covering that cover cells covers the mouth of the archegonium, so called as cover cells. Number 2, they are direct contact with the vegetative cells. Okay, these archegonia are embedded in the thallus, so directly contact with the vegetative cells, no doubt. Number 3, archegonium consists of a neck and a swollen venter. Basal part of the archegonium is the flask-shaped structure. Basal structure of the basal part of the archegonium called as the venter, and upper part of the archegonium is called as the neck. Neck and venter, these are the two main parts of the archegonium. Okay. Number 4, the nearly matured archegonium has 4 cover cells. Cover cells are nothing but the covering cells which covers the mouth of the archegonium. They are 4 in number. 4 to 6 neck canal cells. The neck canal cells number is the 4 to 6, and 1 venter canal cell and 1 egg cell. You see the diagram or photograph of the matured archegonium. At the tip 4 cover cells are there, then 4 to 6 neck canal cells are there. At the venter the single venter canal cell is there, and at the base in the venter there is a egg cell, and the egg cell having the female nucleus, and this egg is nothing but the female gamete. So what is the use of archegonium? It is a male reproductive structure. It develops or it produce or it having the male gamete or egg which is the haploid having the single female nucleus. What is the use of antheridium? Antheridium releases the sperms. They are male gametes. They are haploid having the male nucleus. And what is the function of archegonium? They are female reproductive organ, produces the egg. Egg is nothing but the female gamete, and egg is haploid, contain the haploid female nucleus. So in this way, this is the structure of the archegonium. Then fertilization. Point number 1. Prior to fertilization, the cover cells present at the tip of the archegonium separates. In young condition, cover cells covers or close the mouth of the archegonium, and at the time of fertilization, cover cells can be separate from their union. Number 2, the neck canal cell and venter canal cell disgenerate or disintegrate, and at the same time forming mucilaginous mass. Okay, the archegonium contain neck canal cells, 4 to 6 neck canal cells and single venter canal cells. These cells disintegrate and mucilaginous substance is formed. Okay. Number 3, the mucilage collects in the form of mound at the tip of the archegonium. The mucilage is nothing but the mound-like substance or sticky substance which comes at the tip of the archegonium, okay, at the mouth of the archegonium, and that mucilaginous substance attracts the sperms, male gametes. Okay. Number 4, free swimming antherozoids are attracted towards the neck of the archegonium by the chemical present in the mucilage, and this due to mucilaginous substance the sperms are attracted toward the archegonium. Okay. Then with the help of film of water, with the help of water, sperms are reaches up to the mouth of the archegonium. Number 5, although many antherozoids entered in the neck, many antherozoids, many sperms are entered in the egg, but only one them fuses with the egg and thus oospore is formed. At a time, many sperms are entered in the archegonium, but only one fuse on the basis first come first serve basis. The only single sperm or a single male gamete fuses with the single female gamete and diploid zygote is formed. That zygote is called as the oospore. So in this way, fertilization takes place in the anthoceros. Haploid male gamete sperm, haploid female gamete egg, both fuses and diploid zygote, diploid oospore is formed. And in post-fertilization changes, the growth of the oospore, growth of the zygote takes place, and that growth of the zygote or growth of the oospore produces the sporophytic structure in the anthoceros. Okay. Now the next aspect is the sporophyte. Point number 1. The sporophytes are linear. These sporophytes are developing by the growth of the zygote. Okay. The sporophytes are linear, 2 to 3 centimeter long. You see in the diagram of the matured anthoceros thallus having the upper dorsal side, lower ventral side, and from the dorsal side many sporophytic linear erect structures are developed. Horn-like structures are developed. These linear structures are called as the sporophytes. They are linear, 2 to 3 centimeter long. Then they are elongated structure arising from the dorsal side of the thallus. So in this way, many sporophytes are developing from the single thallus of the anthoceros. Okay. Number 2, the base of each sporophyte is enclosed by a involucre, which is made of gametophytic plant body. At the base of the sporophyte, which is there is a supportive structure to the sporophyte, which is called as the involucre. Okay, this is about the sporophyte. Now the internal structure of the sporophyte. What is the main aim of the sporophyte? Main aim of the sporophyte is to produce the spore. When spore producing structure developing on the plant body, that plant body is called as the sporophytic plant body. Basically, in bryophytes, the gametophytic plant body is the dominant, who produces the antheridium, archegonium. Both are fuses, causes the sexual reproduction in the archegonium, and at the position of archegonium, the sporophytic structure is develops. At that time, that gametophytic plant body named as sporophytic plant body, because spore producing structure is developing after the fertilization. Now the internal structure of the sporophyte. Externally, we cannot find the spores of the anthoceros. For that purpose, you should go through the anatomy of the sporophyte, either TS or LS, then you can observe the spores of the anthoceros. Okay. Point number 1. The mature sporophyte shows a lower foot, middle meristematic zone and upper capsule. You see in the diagram number A. Okay. First point is the thallus cells. Above the thallus cells, there is a lower foot. Foot is the basal part of the sporophyte. Then there is a meristematic layer. Above the foot, there is a meristematic layer. That meristematic layer having the many cells. So this is the intermediate between foot and capsule, and upper linear structure is the capsule, and upper elongated linear erect structure is the capsule. In between capsule and foot, there is a in between foot and capsule, there is a meristematic layer. Okay. Basically, foot, seta and capsule, these are the three parts of the sporophyte in bryophytes, but here seta is absent, and instead of seta, the meristematic zone or meristematic cells are present. Okay. Number 2, the foot is bulbous. असं bulbous तुम्हाला दिसतंच ते तुम्ही व्यवस्थित पहा. The foot is bulbous and is deeply rooted in the gametophytic tissue, and which is deeply rooted in the gametophytic tissue. Number 3, the seta is absent, and instead of the seta, the meristematic zone is present. Okay, we already discussed this. Number 4, in the center of columella, in the center is columella composed of 16 vertical rows of the cells. You see in the capsule at the center there is a columella. Columella is given here, the name columella, which is the central strand of the sporangium, or sporangium means capsule. What is the use of columella? It is the central axis, central strand, gives the mechanical support to the body of the capsule, okay, and which is made by 16 vertical cells. Number 5, surrounding the columella is a cylinder of sporogenous tissue which produce spores and pseudoelaters. Toward the outer side of the columella, the space is present. That space is nothing but the region of sporogenous tissue, and this sporogenous tissue previously they are diploid. Why diploid? Because they develops after the zygote, develops from the diploid zygote or diploid oospore. So these sporogenous cells are the diploid cells, and in sporogenous cells the meiosis reduction division takes place. In this way, many haploid spores are develops in the sporangium or capsule. Okay. Number 5, surrounding the columella is a cylinder of sporogenous tissue which produces spores and pseudoelaters. These elaters are pseudoelaters. They are useful for dispersion of the spores. Okay. Number 6, each spore is uninucleate. Now each spore is the uninucleate, which is each spore is the haploid structure. Why haploid? Because these spores are developed after the meiosis. Meiosis takes place in the sporogenous tissue present in the sporangium or capsule. Okay. Number 6, each spore is uninucleate, haploid, and has a thick ornamental exospores and thin endospores. Exospore outer and thin endospore inner. So each spore is biwalled, having 2 walls, exospore outer and thin endospore inner. Number 7, pseudoelaters consist of 2 to 3 cells joined end to end and a simple or branched structures. So these are the pseudoelaters. They are simple structures useful for dispersion of the spores. Number 8, on germination, spores produce new gametophytic plant body of the Anthoceras. When this sporangium or capsule is matures, it will ruptures, and after rupturing, spores are dispersed through the wind, and when they settle down on the suitable substratum under favorable condition, they absorb the moisture, they will swell up. After swelling, they start germination, and after germination, they produce new individual plant body of the Anthoceras. Now, alternation of the generation. In alternation of generation, upper part is nothing but the vegetative reproduction. Vegetative reproduction in the Anthoceras caused by death and decay of the thallus, then by tubers, by adventitious branches, many types, or by gymi, by many types. The vegetative reproduction takes place. Now the sexual reproduction, that is the basal part of the diagram or life cycle. You see toward your right hand side, male and female 2 names are given. Matured Anthoceras means which is the gametophytic plant body. Gametophytic plant body is the dominant in the life cycle of bryophytes. So we should consider first the gametophytic plant body. So first is the gametophytic plant body, which is the haploid plant body, produce the anthedium as well as the archegonium. Okay, archegonium contain the egg, haploid egg, having called as female gamete contain the female nucleus, and Anthridium produces the antherozoids, sperms or male gametes having the male nucleus. Egg is the non-motile structure, but sperms are or male gametes are motile structures. Now the sperms are attracted toward the archegonium and the fuses with the egg present in the archegonium. In this way diploid zygotic condition is formed. Okay. Now in this way oospore or zygote is formed, which is the diploid structure, and then embryo is formed from the oospore or zygote, and from that small embryo, from that inconspicuous embryo, the sporangium or sporophytic structure is develops. Okay, and sporophytic structure contain the basal foot, middle meristematic zone, and upper capsule. These sporophyte is the linear structures, erect structures embedded in the thallus, and this young sporophyte contain the young sporangium tissue. That sporangium tissue is the diploid structure, goes in the meiosis, and after meiosis they produce many haploid spores, and these when spores are germinates, again they produce the new individual thallus of the Anthoceros. In this way, this is the very simple very simple alternation of generation in the Anthoceros. Then this is the reference. I have used this book, textbook of botany, diversity of microbes and cryptogams, written by Singh, Pandey and Jain, while preparing this PowerPoint presentation. Thank you.